Good evening, everyone. Welcome to this ICRS Basira webinar entitled Discovery: How Gene Therapy is Transforming Osteoarthritis Treatment. My name is Yvan Rotin. I'm professor of pathology, physical therapy, and rehabilitation at the University of Liège in Belgium, and past board member of ICRS. And I will have the pleasure this evening of serving as your moderators. As you know, osteoarthritis affects millions worldwide, and for decades, our treatments have focused mainly on symptoms relief. Today, we explore something different: how gene therapy may redefine the future of disease management by targeting the biology of osteoarthritis itself. To treat this subject, we are very pleased to have three outstanding speakers with us: Dr. Kilian Goose, Professor Chris Evans, and Mrs. Pianella. Together, they will guide us through the science, clinical translations, and practical development challenges shaping this rapidly evolving field. This webinar is supported by an unrestricted grant from Basira, for which we finance for enabling the scientific exchange. Before beginning this webinars, I would like to remind you some practical points. Please submit your questions exclusively through the chat functions. After each presentation, I will select 2 questions for the speakers. If time allows, we will address additional questions at the end of these 3 presentations. I wish you engaging and inspiring webinars. I'm now pleased to introduce the first speaker, Dr. Kilian Kues, who is the Vice President of Genetic Medicine Platform at Basira Bioscience, a NASDAQ-listed leader in innovative non-opioid pain therapies. With over 20 years of experience in gene therapy, he currently leads Basira Germany divisions focused on high-capacity gene therapy vectors. He joined Basira following the acquisitions of GQ Biotherapeutics, a company he co-founded and led as a CEO. We are delighted to have him with us and look forward to his insight today. Kilian, the screen is yours.
Great. Thanks a lot, Yves, for the kind introduction. Okay, so our company, Basira, as mentioned, is a US stock market listed company, so that's why we have these forward-looking statements. And the disclaimer is that this presentation focuses on a pipeline drug candidate currently undergoing clinical testing. The safety and efficacy of this drug candidate have not been fully evaluated by any regulatory authority, including the US FDA and the European Medicines Agency for any indication. And the disclosure is that I'm an employee of Basira Germany, GmbH, which is a subsidiary of Basira Biosciences, Inc. All right, so in gene therapy, and I would actually define gene therapy as the transfer of genetic material into target cells. One of the biggest challenges is how do you actually do that efficiently, the transfer of genetic material into the target cells? And using naked genetic material, DNA, RNA, does not work very well. That's why most groups are using vehicles. We call them vectors. And they basically fall into 2 groups: the ones that are based on viruses and those ones that are not based on viruses. And so among the viral vectors, the most commonly used ones are currently adeno-associated virus, AAV. And I think Professor Chris Evans will talk about that more. Then there are adenoviruses. They have been used in the past quite a lot, but recently they have fallen a little bit out of favor. That's because there were some challenges with these vectors in the earlier generations. We're using the latest generation, which we call high-capacity adenoviruses, or HCAD, also known as health-dependent adenovirus or gutless adenovirus. Then there are also herpes viruses. Some groups are using herpes viruses. And then there are lentiviruses and retroviruses, which are actually integrating viruses. They're nowadays are mostly used for ex vivo gene therapy, so to prepare cells for cell therapy, not so much anymore for in vivo gene therapy. The other viral vectors that I mentioned are actually non-integrating viruses. Now, all of these viral vectors have actually also in the past been used in the context of osteoarthritis gene therapy. And then there are also non-viral vectors, for example, nanoparticles. Most popular ones are the lipid nanoparticles, which became famous because they were the vectors used in the mRNA COVID-19 vaccines. And then there are liposomes and microspheres. Generally speaking, at least to date, the non-viral vectors are not as efficient in transferring genetic material into target cells compared to the viral vectors. That's why, at least nowadays, most groups are actually using viral vectors. All right, so we're using high-capacity animal vector, or HCAD. They look like it's shown here. It's a very symmetrical capsid, icosahedral capsid, so 20 equal surfaces. It's totally made of protein, the capsid. There's no lipid envelope like other viruses have. It's about 100 nanometers in diameter and can carry 30 to 36 kilobase pairs of double-stranded DNA. In comparison to that, an AAV vector is much smaller, as shown here. It's about 20 nanometers in diameter, 4.7 kilobase pairs with single-stranded DNA. Many groups are using double-stranded DNA vectors, so you can only package half, about 2.4 kilobase pairs in those. So HCAD vectors have the advantage that we can transfer up to 30 kilobase pairs of transgene DNA. So that includes then large genes and also potentially multiple genes. What's shown here is a possible configuration of a vector that could, for example, carry 4 different genes in a single vector. And then those genes could also be driven by different promoters. Another advantage is that we have fast onset of gene expression. So depending on the cell type that you're transducing, the peak expression can be as early as 24 hours after administration. And then importantly, we have high transduction efficiency when applied locally. And that in turn means that we only need to use relatively low doses. And just to give you an example here, one approved gene therapy is called Hemgenix. That's an AAV vector expressing factor 9 for treatment of hemophilia B. It's an IV, so injected, so systemic gene therapy. And the dose there is 2 times 10 to the 13 virus genomes per kilogram body weight. So in a normal adult patient, you're somewhere in the 10 to the 15 virus genome total dose range. In the clinical trial that I'm going to talk about, we had doses that ranged from 10 to the 10 to 10 to the 12 vector genomes total dose. So that means we're approximately 1,000 to 100,000 fold lower in doses that we use here compared to the Hemgenix gene therapy. So a lower dose also obviously means a better safety profile, and importantly, it also means that we have relatively low manufacturing costs per dose, which is very important when you're developing a gene therapy for a common disease like osteoarthritis. Also, again, for comparison, the Hemgenix approved gene therapy that I just talked about, that has a list price of $3.5 million per dose. That's something that we cannot do for an osteoarthritis gene therapy. We need to be in a much lower cost level here. So this is very important. Also, our data and data from other groups actually suggest that redosing with HCAD vectors may be possible. And I'm also going to talk about that a little bit later. All right, so we've done a lot of work with the HCAD vector platform for joint gene therapy. Just to show you a few examples, on the top left, we injected mouse joints with HCAD vectors expressing GFP in comparison to different serotypes of AAV. And we found that with the HCAD vectors, we have much higher gene expression in the joints, mostly coming from the synovial tissue, as you can see here in the bright field sections for the corresponding pictures. Then we also confirmed that in rat joints that were injected with the HCAD vector expressing LAG-Z. So that's a marker gene that it can stain blue, and you can see that you get very nice staining of the synovial tissue. In some cases, it seems to be the entire synovial tissue that is actually stained. So very efficient transduction into the synovial tissue with these vectors. Into the chondrocytes, the vectors do not go very efficiently. You have some transduction in the superficial chondrocytes, but the vector is not really able to penetrate into deeper layers of the cartilage. And then we also confirmed long-term gene expression. We injected mice into the joints with a HCAD vector expressing luciferase. That's an imageable marker gene, and we found that you have an initial drop of gene expression, which is typical for adenovirus-based vectors, but then you have a stabilization at a certain level, and the gene expression levels remained at that level for the entire period of the experiment, which was a little bit more than 1 year. That's when the mice started to die of old age, so we had to stop the experiment here. So essentially it was in mice lifelong expression. So very important, we have efficient transduction of the synovial cells in the joint, and we have long-term gene expression. So switching gears a little bit, interleukin-1, IL-1 is a major driver of chronic inflammation and disease progression in osteoarthritis. It is overexpressed in most osteoarthritis joints, and it drives the expression of MMPs, so enzymes that degrade cartilage, and cytokines like interleukins and TNF-alpha that promote the inflammation in the joint. So we have bone remodeling, cartilage degradation, and pain that is really driven by interleukin-1. Now, there have been a number of studies that showed that by blocking interleukin-1, you can actually treat osteoarthritis quite well. So these are two examples. On the top, these were dogs with osteoarthritis induced and then treated with recombinant interleukin-1 receptor antagonist protein. That's the endogenous inhibitor of interleukin-1. And you can see that you had improved cartilage lesions in the treated joints that was also dose dependent. And then on the bottom, we have a clinical trial. There was actually a very big clinical trial with more than 1,500 patients. It was not a clinical trial for osteoarthritis, but it was for a cardiovascular indication. And these patients were treated with a monoclonal antibody against interleukin-1. And it was found that the patients in the treatment group had significantly lower rates of knee and hip joint replacement. So that was also a very good proof that on the one hand, interleukin-1 plays an essential role in driving OA disease progression, and blocking IL-1 can effectively treat osteoarthritis. So PCRX-201, our drug candidate, is a high-capacity adenovirus vector expressing interleukin-1 receptor antagonist, so the endogenous inhibitor of IL-1. It enters mostly the cells in the synovial tissue, and it has a inflammation-inducible promoter. So it's activated by NF-kappa B, which is present in inflammatory conditions in the joint. And then you have the production of the protein interleukin-1 receptor antagonist, IL-1RA, that's secreted into the synovial fluid and then binds to the interleukin-1 receptor on the surface of the cells and blocks it and prevents signaling through IL-1 and therefore slows the progression of the disease and inhibits the inflammation. So it's a very nice tunable mechanism that we actually have due to the inflammation-inducible promoter that we're applying here. So we did a lot of preclinical studies in various different animal models for osteoarthritis. Just to show you a few examples, this is an experiment in the mouse cruciate ligament tear model. where we analyzed by micro CT and found that in the joints treated with the R1A vectors, so analog to 201, we had significantly higher cartilage volume, significantly higher cartilage surface, significantly higher bone area covered by cartilage, and we did not find osteophytes, whereas in the control groups, we had actually osteophytes present. Then another example here, a horse model of osteoarthritis. So this is an osteochondral fragment model. We found a significantly improved histology score and also significantly improved functional scores looking at lameness, range of motion, flexion, and diffusion. So these and other encouraging results for the 201 vector then led to the initiation of a phase 1 clinical trial. And that enrolled 72 knee osteoarthritis patients with a KL grade 2 to 4. There were 2 cohorts. The second cohort was pre-treated with a corticosteroid, so it was a injection of methylprednisolone immediately before 201 administration, also intra-articularly, and cohort 1 was not pre-treated. And we had doses of 1.4 times 10 to the 10 in the low dose group, and then going up to 10 to the 11 and 10 to the 12 virus genomes total dose. The endpoints were obviously safety and tolerability, and then we had exploratory efficacy endpoints, which I'm going to talk about in a minute. So importantly, 201 was safe and well tolerated across a 100-fold vector dose range. There were no serious treatment-emergent and adverse events, and the most common treatment-related adverse event was joint effusion, which was actually more common in the non-pre-treated group and less common in the group in the cohort that was pre-treated with the corticosteroid. Yeah, and then looking at the WOMAC-A pain score, we had a marked improvement compared to baseline in all of the dose groups, and that was actually sustained throughout 3 years at a very stable level. And when we look at the pre-treated versus the corticosteroid non-pre-treated group, we actually found that in the pre-treated group, we had an even better effect with about 50% reduction compared to baseline, whereas in the non-pre-treated group, it was 38 to 36%. Yeah, and a similar picture with the WOMAC-B stiffness score. Also here, marked improvement that was sustained throughout 3 years and was actually true for all of the dose levels. And also here, the cohort that was pre-treated with a corticosteroid had a better improvement compared to the non-pre-treated group. Yeah, and then we also looked at neutralizing antibody and the impact that might have on the treatment effect. And we actually found that in the patients, whether the patients had pre-existing antibodies against 201, which is based on the adenovirus serotype 5 capsid, whether they have antibodies or not, did actually not have an impact on the treatment effect. And that was true for both cohorts. So that actually suggests that it may be possible to retreat patients, either retreating the same joint or treating patients' other joints that may also be affected by osteoarthritis. Because if patients do not have neutralizing antibodies at the outset of the first injection, they will most likely have them after the first injection induced by 201. But retreatment, according to this data, might actually be possible. All right, and then so these encouraging results then led to the initiation of a phase 2 clinical trial that's currently that was initiated. It's a 2-part trial. We have subjects with painful osteoarthritis of of the knee with a KL grade 2 to 4 that are being enrolled. And both cohorts have 3 groups, the low dose and the mid dose group from the phase 1 clinical trial, and then a placebo group. And all of these groups get a corticosteroid pretreatment, as was also done in the phase 1 clinical trial. The difference between part A and part B is that in part A, we're using 201 material that was produced with the same manufacturing process as in the phase 1 clinical trial. And in part B, we're using material that is produced with a new, more scalable manufacturing process that would then later also be the commercial manufacturing process. Right, so yeah, this trial with part A was initiated earlier this year, and we're looking forward to getting top-line results at the end of this year. Yeah, and with that, I'll stop here and happy to take questions.
Thank you very much, Kilian, for this very interesting tour and sharing with us these very promising results. The next speaker is Professor Christopher Evans, and it's very great honor for me to introduce Christopher. Christopher is the John and Posey Credible Professor of Orthopedics at the Mayo Clinics, and holds professorships in physical medicine and rehabilitation, orthopedic surgery, and molecular medicine at the Mayo Clinic College of Medicine and Science. He earned his degrees in genetics, microbiology, and biochemistry at the University of Wales, followed by postdoctoral research in molecular biology in Brussels, in Belgium. In addition to his scientific training, he holds a master's degree in the history and philosophy of science from the University of Pittsburgh, and is professor emeritus at Harvard Medical School. He is also the co-founders of Jena Sense Corporations, an arthritis gene therapy company. We are very pleased to have him with us today, and please, Christopher, share your slide deck.
Right, thank you, Yves, and thank everybody for logging in. So I've been asked to speak on this topic, but before I do, I should let you know my one disclosure, which is that 4 colleagues and I founded an arthritis gene therapy company called Genescience, which obviously overlaps with the present talk. So just like Dr. Goose, we're interested in delivering the interleukin-1 receptor antagonist to joints with osteoarthritis for the same reasons that he already cited. But to get it there, we're using a different vector. We're using the adeno-associated virus, which we like for several reasons. Some of them are listed here. But a fundamental one is that AAV causes no known human disease. And for human gene therapy, especially for a non-lethal disease, the safety issues are paramount. It's small, and as I'll show you in a few slides' time, it gets into the articular cartilage. And also it's been approved by the FDA for human clinical use. And last time I checked, there were 8 human gene therapies approved by the FDA that use AAV as the vector. With this in mind, we made ourselves a vector that's shown here. It's serotype 2.5. And as the previous speaker said, the wild-type virus is single-stranded DNA genome. The self-complementary AAV is configured to have a double-stranded genome, which means it's much more efficient. The transgene IL-1RA, the cDNAs have been codon optimized. That's what CO stands for. There's an SV40 polyadenylation signal, and translational efficiency is further enhanced by a Kozak sequence and an intron in the 5' untranslated region that stabilizes the RNA. And it's driven by CMV, cytomegalovirus enhancer promoter, that's quite popular in gene therapy because it's a strong constitutive promoter. As I'll show you in a few slides, our preclinical data suggests an element of inducibility that's interesting and potentially useful. And we've taken this through rats, rabbits, and horses. Obviously, don't have time to go into them all in a short talk, but some of the insights that I've been asked to talk about are best manifest in the horse studies. These were conducted by my colleague Steve Givazani at the University of Florida that has a veterinary school. And what Steve did was put the green fluorescent protein cDNA into our self-complementing AAV and inject it into the joints of random horses that were walking around over in the veterinary school. And 2 weeks later, went in to see where the gene was expressed. The top panels here show synovium that's been taken out of the horse and just put fresh and wet onto the stage of a fluorescence microscope. In the absence of the gene, obviously nothing's green or fluorescent. But after 2 weeks after injection of the AAV, you get beautiful lighting up of the synovial villi shown here in higher magnification. And if you look in cross-section, what you see is that the synovial lining cells are very efficiently transduced, nothing in the subsynovium. And this is shown again in higher magnification. And this is sort of what we'd expected, because we'd seen in previous work that the synovium is very readily transduced by AAV and other vectors. What was a nice surprise was to show that we're getting transduction of the chondrocytes. So if you look at this panel on the top left, this is the joint surface, and all the way through to the deepest zones of the cartilage, we're getting very efficient transduction of the chondrocytes, including, interestingly, in these nests that are characteristic of osteoarthritis. These were adult horses who seem to have some osteoarthritic changes, and we can get right into those cells. And I think that's very important, given that chondrocytes might lay at the epicenter of the pathophysiology of osteoarthritis. So we can take our genetic medicine right into the heart of the disease. The other thing that came out of these preclinical studies that we can get long-term gene expression. Again, this is from Steve's lab in Florida, showing that we can get expression in the joint of the horse for at least a year. And there's some horses now gone out 18 months. So this is where the equine version of IL-1RA has been cloned into our vector. And they're tapping synovial fluid every month and measuring IL-1RA, equine IL-1RA by ELISA. So we're getting long-term gene expression that seems to reflect the importance of the chondrocyte as a depot for this process. So here, the virus was injected into the horse's joint, and at 2 weeks or at 52 weeks, synovium and cartilage was obtained, and viral genomes were measured. And here they're on a per cell basis. Here stands for cellular DNA. You'll see at 2 weeks on a per cell basis, there's nearly twice as much virus genome in the cartilage as there is in the synovium of the horse. If you go out for a year, almost all the synovial expressions disappeared, and it's nearly all in the cartilage. And that sort of meshes with what we know about the physiology of joints. The synovium is a tissue that turns over. AAV doesn't integrate, so as the cells divide, it's going to be lost, whereas the cartilage doesn't go anywhere. It sticks around and serves, we think, as the site of long-term gene expression in the joint. The other thing we saw that was interesting in these preclinical experiments was that in the presence of inflammation, transgene expression, the IL-1RA expression went up. So if you put AAV IL-1RA into a normal horse joint and measure synovial fluid, there's about 50 nanograms per ml. If you do it in a joint that's just undergone surgery and the model of OA in the horse is induced surgically, you get 4 to 5 times more expression than you did at baseline. And this then diminishes as the inflammation subsides. So the other useful thing from the horse data was that it enables you to do some quite clinically relevant testing because they have large joints of human size. And I'm only going to show one in the interest of time. This is a lameness study where the horses are run on a treadmill. There's an instrument called a kinematic analysis lameness locator. It's about all I know about it, but it gives you lameness, which is a surrogate for pain in quantitative units, in this case, millimeters. And a horse that's undergone the creation of an osteochondral defect gets osteoarthritis and it has a certain lameness. After you inject the virus, the lameness goes down. And what's interesting is that it goes down progressively. So at the end of this 12-week experiment, the lameness is still declining, suggesting that this is a progressive process that we don't quite understand, but it's highly encouraging in view of the fact that we wanted to go into clinical trials. So when we approached the FDA, we were able to give them a fairly hefty dossier showing that we could deliver IL-1 R8 in various different animals, that we got sustained transgene expression in a dose-dependent manner. I didn't show you the dose response. The horse model's been run out acutely and chronically, and the FDA sent us away to do a very large safety study in rats, which enabled us to get investigational new drug, IND approval, which enabled us to go into a clinical trial. And for those of you that are interested, this is the identifier on clinicaltrials.gov. The structure of the trial is shown here. It's dose escalation going from 10 to the 11 viral genomes in 3 patients, 10 to the 12, and 10 to the 13. The key inclusion criteria was Kellgren-Lawrence score of 2 or 3, moderate osteoarthritis, and the FDA required us to recruit patients who failed at least 2 prior non-surgical therapies. Being a phase 1 study, the primary endpoint, safety and tolerability. But while we were doing it, we did a whole bunch of secondary endpoint analyses, and they're listed here. And the study ran for a year with serial office visits from these subjects in the study so we could track progress. This is the distribution of the subjects that are in the trial. Twice as many females as males, which is typical for osteoarthritis. We had an upper BMI limit of 40, so all patients were under that, and typically middle-aged, and the entry criteria required them to have 2 or 3. In terms of the adverse events, what you don't want in a study like this is a serious adverse event that's related to the drug and it doesn't get better. So the first patient, low dose, broke her foot. This was serious, but it wasn't related to the drug, and the bone healed unproblematically. And then complained about stiffness in the hand that was not serious and was unlikely to be related to the drug. And we don't know what happened because at 6 months, this individual was under COVID lockdown in a foreign country. So we weren't able to see what happened. And the same in the second low-dose patient, dyspepsia, hematoma. It's very difficult to get blood from this patient and often resulted in a hematoma. And she walked into some barbed wire at a certain stage, all unproblematic concerning the drug. In the mid and the high dose, patient 4 in the mid dose had a knee infusion after injection of the virus. Now, this patient had a squishy knee to begin with, so we don't know whether it was because we injected the virus or was just part of the natural history of her joint, but it resolved with rest and ice. The other mid-dose patient, number 5, complained of knee pain in between doses, in between office visits, so we don't really know what was going on, but it resolved by the time of the second office visit, so it was unlikely to be drug-related. And patient 9, the last patient in the high-dose group, had an infusion that was possibly related. It occurred after the injection, but got better with ice and rest. So there are no serious adverse events in that score. And we did a comprehensive blood count and chemistry, and none of these measures were affected by injection of the virus. And we were particularly pleased to see there was no neutropenia, because neutropenia is a recognized side effect of injecting recombinant IL-1RA. It's the drug anakinra, and if it's delivered systemically, it gives neutropenia. We were reasonably confident we wouldn't see this, because in all of our animal models, even when expressing high amounts of IL-1RA in the joint, we've never seen it elevated in the peripheral blood, and we didn't see it here either. In terms of gene expression, the patients who got the 10 to the 11 dose, subject 1 got a very nice textbook increase in IL-1RA in the synovial fluid, then went into lockdown, so we couldn't see what it was like at 1 year, 52 weeks. Patient 2, as well as having invisible veins, had no synovial fluid that we could extract. It was a dry joint. But patient 3 did very much similar to what patient 1 did, and at 1 year was still expressing. In the mid dose, now these y-axes have different scales, so you should be aware of that. The patients 5 and 6, they went up after injection of the virus and stayed up for the whole year. Patient 4, who had the effusion, had a huge amount in the synovial fluid at 1 month, but then it came back down and actually settled at a lower level than that of the other 2 patients. We're not quite sure why, but that's the observation. And at the highest dose, patient 7 went up and stayed up. The other 2, patient 9 had the effusion, and just like patient 4, had a big spike in IL-1RA expression, and then it came back down again. And patient 8 did something similar. And panel B shows the averaged amounts. In terms of what happened to the patient-reported outcomes, patients came in with a range of pain scores on a numeric rating scale, and the basic message is all went out with a less pain than they come in, some dramatically. Patient 6, for instance, had a pain score of 7 out of 10 and was pain-free at the end of it. Some patients, well, one patient had no pain coming in, but had no pain going out, which was nice because it showed that what we were doing was not causing any symptoms that they didn't have initially. And the WOMAC scores, the function scores, really reflect what we'd seen with the pain scores, as they came down modestly in the low dose group and quite dramatically, for instance, again, patient 6 in the high dose group. So we concluded from all of this that the treatment was safe, the patients tolerated it. Some wanted to have their other knee done, but of course we weren't allowed to do that. Increased expression in the 8 subjects where we get fluid that was maintained for a year, but it fell off peak values after an infusion. Didn't show data on this, but less than 1% leaked out in the peripheral blood, and that was gone by a week. The neutralizing antibodies, that's well known for AAV, but no cell-mediated response, and there was a trend towards improved pain and function. There's been a follow-up 1B study by the company we set up, Geneson's. Larger study is basically to distinguish between the medium and the high dose as the dose to go forward with, given they were superimposable in phase 1, and included groups getting steroid to see if this had effect on expression and antibody response to the virus. 1 year follow-up, it's been completed, and the first look at the data confirmed safety, confirmed gene expression, and confirm clinical responses. This had a placebo group that we didn't have in phase 1. So the next steps are to get into a pivotal study. So the FDA has given this fast track designation and also designated it an RMAT procedure, and it's cleared our phase 2/3 protocol, so we can initiate enrollment in this pivotal trial, which is something we're trying to do as fast as we can. So I'll end by thanking the people that funded this. The preclinical work was funded by NIH and the US Department of Defense, as well as Mayo Clinic. And phase 1B was funded by California Institute of Regenerative Medicine. I'd like to thank my co-workers. Myself, Steve, and Paul Robbins have worked on this for a long time, as you can see here when we were brought together in Pittsburgh. We've meandered off in different directions, but stayed tight. And the data I've just shown you are as much theirs as they are mine. I'm really just spokesman. Thank the clinical trial team at the Mayo Clinic, which is absolutely fantastic. It's difficult to do these studies, and you need lots of different expertises. And I'd like to thank Tom Chauberg and Ida Caravella at Genesont, who were instrumental in getting the vector manufactured. With that, I'll stop. Thank you very much.
Thank you very much, Chris. I suggest now we move to the next and last speaker, the woman representative of this webinar, Monica Pianella. Monica is a regulatory affairs specialist with extensive experience in advanced therapies. She holds a master's in pharmaceuticals chemistry and technology from Sapienza University of Rome, and has contributed to regulatory best practice through a secondment to the Medicine and Healthcare Product Regulatory Agency, ATMP Working Party. She has worked in cell and gene therapy since 2016, starting at GSK before moving into regulatory roles at the Cells and Gene Therapy Catapult. She is currently a Director of Regulatory Affairs at Boyce Company, where she supports biotech companies and research organizations with clinical development and regulatory strategy. Please, Monica, the screen is yours.
Thank you. Thanks for the introduction. Good afternoon, good morning, everyone. I'm very pleased to say we've now reached the final part of this presentation, but I'm more than happy to continue this conversation. So we are going now to talk about regulatory considerations when it comes to developing gene therapies for cartilage repair, with particular focus on osteoarthritis, to continue what has already been introduced by the previous experts. So we are very well aware of the articular cartilage role, which is to preserve mainly joint structural integrity, but also we are familiar and we understand that articular defects can have limited ability to regenerate. And amongst the conditions which affect the articular cartilage function and structure, osteoarthritis represents by far the most common and most impactful condition. And it still represents, as of today, a large unmet medical need, because as has been presented previously, current osteoarthritis pharmacological treatments are mainly aimed at addressing and managing symptoms. They have limited effectiveness, and they may also lead to significant adverse events, especially when used in the long term. And most of all, there is no disease-modifying drug currently approved. And this gap is further enhanced by patient surveys, such as the Global Osteoarthritis Patient Perception Survey, where more than 40% of patients declare of not being satisfied with the current way treatment. And this is where exactly the gene therapy could play a transformative role. And for example, gene therapies such as vector-based gene therapies, and as some examples has been given previously, adenoviral vectors or AAVs. And this is because they may offer sustained local expression of therapeutic proteins, but they may also offer potential true disease modification. So let's now take just a quick step back to understand how these types of therapies are regulated. So in the European Union, gene therapies alongside cell therapies fall under the category of advanced therapy medicinal products, or ATMPs, and they're regulated under the Regulation 1394/2007. And they are very clearly classified into 3 or 4 subcategories, including gene therapies, somatic cell therapies, tissue engineered products, and combined NTP, where, for example, there is a medical device component, which is an integral part of the product. In the United States, instead, the corresponding and broadly corresponding regulation is Title 21 of the Code of Federal Regulation 1271. However, this is more focused on also providing a definition of human cells and tissue-based products. There is no such a clear classification in the US jurisdiction. Therefore, guidances are issued by the FDA to address these subtleties between cell and gene therapies. And from a clinical trial perspective, usually trials are assessed nationally, and with the implementation of the EU Clinical Trial Regulation, or UCTR, there is now a more harmonized assessment process and submission which happens through a common portal. In the United States, instead, the regulation applicable for clinical trial is captured in Title 21 CFR 312 and 56, respectively for IND applications and institutional review boards. But in addition to these standard and well-known approvals, which are required before initiating clinical trials, there may be also the need to additional biosafety approvals for some categories of these products. For example, in the European Union, this is regulated under the genetically modified organism legislation, and it is very worth mentioning that it may be a very lengthy process and can have an significant impact on clinical trials and sites that have timelines. So it's highly recommended to factor these in the program and plans for approvals, to obtain approvals. Now, moving on to talking about development consideration within the context of regulatory expectations, and we're going to briefly touch base on non-clinical, quality, CMC, and clinical. And in doing that, in generating a development package, it's always important to remember that in the context of osteoarthritis, this is not considered a life-threatening condition. And therefore, the regulators will expect a much tighter risk-benefit profile of the product, and the evidence that we will generate will need to reflect this higher standard. So starting from non-clinical, so whilst for small molecules and non-clinical data package is relatively clear, this clarity becomes less or more faded as we go into biologics and especially ATMPs. This is because the nature and the risk profile of the product will drive the type of data set that will need to be generated. So from a primary pharmacology perspective, in vitro and vivo proof of concept studies are expected. And it's very important to mention here the choice of the animal model, where, especially in terms of the disease animal model, because it should ideally represent as close as possible the pathophysiology and disease stage of the patient. From a pharmacokinetic perspective, the typical ADME data package is not really relevant for ATMPs, but the focus is more on biodistribution studies and vector shedding assessment, with the latter normally needed for environmental risk assessment. And it's very important to remember that information which are publicly available, previous use of similar vector-based gene therapies in humans and clinical trials will be relevant to support the risk assessment for your product when it comes to vector shedding. So it's highly recommended to do the literature search before looking into along with looking into the risk associated with your product. Now, from a safety perspective, the typical safety pharmacology core battery of studies is not needed for ATMPs really, because usually endpoints are included in in vivo toxicology study. And when it comes to in vivo toxicology, this is what's usually defined as pivotal non-clinical safety study. And as such, it's expected to be conducted to GLP unless otherwise justified. And it is also quite common to see a combination of safety and biodistribution assessments within the same study, also to save on animal use. And it's also very important to include immunogenicity endpoints as part of the study. Now, genotoxicity studies are not usually applicable to ATMPs unless there is any specific concern around particular process-related impurities or components of the delivery system. Instead, the focus is mainly made on assessing the integration profile of the product to address the insertion and mutagenesis risk. And also in this case, information which is publicly available with similar vector-based gene therapies can also be used to help you in this risk assessment. From a reproductive toxicity perspective, biodistribution of the vector to the gonads is usually included in the biodistribution study. However, if it is anticipated that there may be some effects on the reproductive function and development, then these studies would be expected to be conducted. However, this is not necessary before first in human, but usually before exposing larger patient populations to the therapy. Now, from a quality or chemistry, manufacturing control perspective, we know that CMC is a major regulatory hurdle for gene therapies, let alone for gene therapies developed for osteoarthritis, because, for example, unlike gene therapies for rare disease, there is an additional challenge brought in terms of scaling, because, of course, the manufacturer will need to manufacture the therapy to meet a much larger patient population. And one important point I wanted to make, because we could spend a whole hour to talk about as well CMC considerations. One point I wanted to make is also the importance of aligning process development with non-clinical and clinical development in order to limit as much as possible changes mid-development, because they might trigger the need of additional bridging studies, leading to further delays and more resources. And finally, from a clinical perspective, in preparation to the exploratory trials, we know that in gene therapy, it is not ethical to test the drug in healthy volunteers. Therefore, exploratory trials will start with patients directly. And that's also the reason why the starting dose must be selected not only because safe, but also because it is expected to have a pharmacological effect. And now, from a population perspective, there are multiple considerations to take into account, especially in the context of osteoarthritis. But a couple of points that's relevant to make is around the consideration for pre-existing immunity to the IMP, given the immunogenicity profile of especially vector-based gene therapies, and also homogeneity of the patient groups studied in the trial. So, for example, in the context of osteoarthritis, you might start in enrolling patients targeting just a unilateral joint, or you might use this principle to help yourself with patient stratification in the study. And from a study design perspective, for gene therapy, it's quite common to see flexible and adaptive designs where dose escalation phase is followed by a cohort expansion aimed at assessing or identifying recommended dose for subsequent trials. However, it's also very important to remember how to manage potential confounding factors, which might, for instance, derive from the use of concomitant therapies, because these may interfere with the outcome measures. So according to the OA guidance from the EMA, the intention is to limit and avoid as much as possible confounding factors. However, it is unethical to exclude and withhold standard of care from patients. So there is always different approaches that can be used, for example, standardizing the potential need of rescue medications, for instance. And finally, the patients treated with gene therapies are expected to be followed in the long term, especially from a safety perspective. And the duration of these studies are usually defined based on a risk-based approach, considering the nature, mechanism of action, persistence of the product, but also age-specific considerations of the target patient population. And then I would like to conclude with the case study, some lessons learned, which is mainly a summary outcome of a number of agency interactions on the development of vector-based gene therapies for a variety of therapeutic indications, including the treatment of osteoarthritis of the knee. And from a non-clinical perspective, one of the common themes is around dose selection study and also the material used in this dose selection study, because this must be representative of the clinical vector when it comes to vector construct, particularly the promoter used and the gene of interest, but also the relevance of the animal model, as mentioned earlier. Also, in gene therapies, we usually talk about a long-term effect, and also this is valid for osteoarthritis. And therefore, there is an expectation from regulatory authority to provide an explanation on mitigating vector dilution in mitotic cells over time. And also, data is expected from an in vivo long-term expression study to support the claim of long-term effect. From a quality perspective, usually a common theme is the potency assay, and usually surrogate assays are acceptable for first in human, mostly using a matrix approach. but then the functional bioassays, preferably in vitro, needs to be developed early, as early as possible. And in the European Union, there is a much stricter expectation that these activities started very early in development, and if not, a justification should be provided. From an impurity profile, usually it's recommended to avoid using 293 T cell line for vector production, or sponsors would be expected to show absence for the SB40 T antigen sequence, along with a risk assessment and tumorigenicity data. And finally, from a clinical perspective, when it comes to participants' enrollment, in the initial cohorts, at least of a clinical trial, you should enroll patients which are at a more advanced stage of the disease, so they have a more favorable risk-benefit balance. And then as more safety data becomes available, then you will be able to enroll participants or patients with a less severe stage of the disease. And another important point is about pre-existing immunity. So there can be cases where sponsors might decide not to exclude patients with pre-existing immunity, but in that case, it would be important then to balance recruitment challenges, but also the confounding factors when assessing efficacy. And from a study design perspective, we've talked about the flexibility, which is quite common for gene therapy trials. And is usually recommended to set it up from the very beginning in your protocol. However, this flexibility may or may not be acceptable depending also on the disease characteristics. So it's very highly recommended to reach out to the competent authorities, engage them as soon as possible, and really understand what's best to design your protocol. And then finally, just 3 takeaway points that I wanted to share with you as a recap, really. We talked about promising use of gene therapies for the treatment of osteoarthritis, either symptom management and or disease modification. However, we have a higher regulatory bar to meet here, opposed to the development of gene therapies for life-threatening diseases. Also, we talked about the criticality and importance of engaging early with regulators. And finally, I mentioned about the GMO approval processes in the European Union, which may be very lengthy and can have an impact on the clinical trials timelines. However, there are some efforts to improve with the new European pharma legislation and the EU biotech tax proposal, which hopefully will implement a streamlined approach and potentially some waivers for the most known and HMPs. And this will hopefully bring treatments to patients with less administrative delays going forward. And with this, I'm ending my presentation. Happy to take any questions if there is time. Thank you.
Thank you very much, Monica, for this very interesting guidance framework.